So, hello everyone. So, I will be starting with this topic on somatic embryogenesis where we have left. So, before I start with this, see, embryos it is not necessary that they originate from the zygote. Instead, like they can originate from variety of somatic cells, and that consequently is referred to as a somatic embryogenesis, right? So we can write that somatic embryogenesis, it is the process in which a single or group of somatic cells initiate the developmental pathway that leads to the formation of non-zygotic embryos. Or we can say that here the zygotic embryos, they develop from, see, like zygotic embryos, they develop from the fertilized eggs. They are termed as zygotic, right? Whereas the non-zygotic embryos, they develop from the cells other than the zygote. So we can also say that the somatic embryos are bipolar structure. In that, they have a radical and a plumule. And these embryos, they have no connection with the pre-existing vascular tissues within the maternal callus. So we can also say that somatic embryogenesis is a non-sexual developmental process that produces a bipolar embryo with a closed vascular system from the somatic tissues of a plant. So this is brief about the somatic embryogenesis. Embryogenesis can be produced directly or indirectly. So let us start with that. Just a minute. Okay. So stuff, it is not working. Okay. So somatic embryos, they can be produced by two methods. They can be produced directly, or either directly they can be produced or indirectly. If I talk about the directly, this direct somatic embryogenesis, the embryo is formed directly from a cell or small group of cells without the production of an intervening callus. Though some common tissues, usually reproductive tissues such as nuclear style or pollens, right? Direct somatic embryogenesis is generally rare in comparison with the indirect somatic embryogenesis. So we can here write that direct, that is a rare as compared to the indirect one. If I talk about the indirect somatic embryogenesis, here the callus is first produced from explant. Embryos can then be produced from the callus tissues or from cell suspension produced from that callus. So here is a schematic representation of the sequential stages of somatic embryo development. So let me share that also with you. This is a schematic representation of the sequential stages of somatic embryo development. So it is like this. There is a single cell. This can be a single cell or it can be a group of the cells. Repeated cell division leads to the production of group of cells that develop into an organized structure known as globular embryo. Further, the development results in the heart stage embryo Then the torpedo stage embryos, from which the plants can be regenerated. So this is all about the somatic embryogenesis. Hope it is clear. The next topic which I will be taking is the organogenesis. Organogenesis is the formation of organs, either shoot or root. So it is the de novo formation of the plant organs using either a meristematic or a non-meristematic tissues. So the process of organogenesis involves three main phases. So with three main phases which are involved in the organogenesis are: number one, the de-differentiation. Second is the induction. And third is the differentiation. So there are two methods of plant regeneration by organogenesis. One method is the organogenesis through the callus formation, which is, again, it is of two types. There is a direct organogenesis and there is an indirect one. So first I'm talking about the indirect one. Indirect organogenesis. Here, the method of organogenesis is through the callus formation. And then there is a direct organogenesis. This it is directly from the explant. So in the direct organogenesis, there is no intervening callus stage, and root, shoots, and other organs are directly, they will arise from the explant. So the main feature of organogenesis is that it relies on the inherent plasticity of the plant tissue. And this is regulated by altering the components of the medium like that will determine the developmental pathways, right? The pathways of degenerating tissues. So in particularly, if there is a high, this just example I'm giving, in case this is a high auxin, to cytokinins, they will favor the root formation. So the opposite will favor the shoot formation. It means when cytokinins are more than auxins, then it will favor the shoot formation. And intermediate level will enhance the callus formation. So, agar intermediate hota hai in dono ka ratio. So intermediate level will enhance the callus proliferation. So there is a difference between a somatic embryogenesis and organogenesis. The whole plant regeneration versus, the whole plant regeneration where the organogenesis is a monopolar structure. So this will be the monopolar structure. So monopolar structure forms a connection with the pre-existing vascular tissues within the callus. On the other hand, somatic embryogenesis is a bipolar structure because the embryos that are formed will contain a radicle and a plumule, right? So the embryos also, they have no connection with the pre-existing vascular tissues within the maternal callus, all the cultured explants, right? So we can say that induction of a somatic embryogenesis requires a single hormonal signal to induce a bipolar structure, which is capable of forming a complete plant. Like while organogenesis, it requires two different hormonal signals to induce the first shoot organs and then the root organs, right? Okay. So I'll just stop over here. Thank you so much.